Antigens are substances that can be recognized by the immune cells. They can be from the self, they can be foreign, they can be pathogenic, immunogenic, and they're pretty much a broad category. They can also be in numerous, or they can be all macromolecules. The important thing about antigens is that they have this area called the epitope, which is it's a localized region on the antigen, and it's specifically bound by immune cells and receptors in this location. The analog in the antibodies or the receptors is called the paratope. So if this is the epitope, or yeah, if this is the epitope, then this would be the paratope. B cells use BCRs. They don't require a mediator to be present. They don't require a mediator or a helper. T cells, however, do. They do require a mediator. Typically they use MHCs. BCRs, they can bind to just about any type of antigens, whether it be proteins or nucleic acids or lipids. TCRs, however, they can only bind to peptides, which are being carried by MHCs, whether it be MHC1 or MHC2. MHC1 is it presents intracellular antigens which can be activated, which can activate the cytotoxic T cells. So it's intracellular, so antigens have to be inside, which are taken outside by vesicles and placed on the MHCs. Class 2 MHCs are a little bit different in that they're typically in phagosomes or phagocytes, which engulf antigens, and then they take them on the outside, similarly how they do in the MHC1 too. MHC1. And these MHC1s, they're typically found in all nucleated cells, but MHC1s or MHC2s are found on almost in APCs, professional APCs. Their structures are mostly similar, but MHC1 is a little bit lopsided in that it has a longer heavy chain and a short light chain. Class 2 is a little bit more symmetrical and it has two longer chains rather than the asymmetrical chains over here under MHC1. It's also worth noting that they both have binding clefts or the areas where the antigen is binding. Though class 1 has a shorter cleft in that it can bind to only shorter proteins than it would if it were MHC2. In terms of their antigen processing pathways, class 1 has antigens that are made inside of the cell. So if the cell is infected with a virus, it's producing viral proteins. So we can say that these are viral proteins. They will be proteased and turned into peptides, which are little smaller bits. These peptides are taken to the rough ER, I'll draw here, and they are loaded by TAP. TAP will then load them onto MHC1, where they will be placed and where they will wait for the CD8 cytotoxic cell. MHC2 occurs in APCs like B cells. They grab proteins floating around the cytosol and they engulf them into vesicles. They are also broken down by lysosomes, so they turn into smaller pieces. These small pieces, or these peptides, MHC2 then assembles within the cell, and it's taken to the ER as well, with an invariant chain, which protects it from being reactive with anything inside the cell. As the MHC moves outwards towards the plasma membrane, the invariant chain leaves, and now it has a peptide inside of it, and now it can bind to. And now the MHC can be recognized by CD4 and now the MHC can be recognized by CD4.
Dendritic cells are antigen-presenting cells that initiate T cell activation. This can happen when an immature dendritic cell captures microbes using lectin receptors and engulfs them via phagocytosis. Microbial products then bind to toll-like receptors, activating the cell. Once it is activated, it migrates and matures from the tissue to the draining lymph node, where it presents the antigen via MHC2 to T cells. As it matures, it increases MHC molecules and co-stimulators. Dendritic cells also have the skill of cross-presentation, which happens in a dendritic cell when internalized antigens go through the cytoplasm of a dying cell into a dendritic cell, where it is expressed through MHC1, expressing the internal antigens. The structure of the receptors of both B cells and T cells are very different, where the B lymphocyte receptor is Y-shaped, can be secreted, and can recognize micromolecules. In addition to this, it is made up of 4 polypeptide chains, 2 heavy and 2 light, making up for 2 binding sites. The TCR is made up of 2 polypeptide chains that are cell-mediated and cannot be detached, and only recognize peptides making up only 1 binding site. Similarly, both receptors have a constant and variable binding region. The B cell recognizes antigens through their B cell receptors, which can be membrane-bound antibodies that can recognize intact antigens through their fragment antigen binding region. Meanwhile, T cell receptors cannot recognize an intact antigen, meaning it can only recognize an antigen when it is displayed as a peptide by an MHC molecule. The diversity of antigen receptors is generated during the process of lymphocyte development by the initiation of somatic recombination or gene segments that can code variable regions of the receptor. For example, the genes that encode antigen receptors are broken into V, D, and J gene segments instead of being intact in the germline, meaning that during lymphocyte development, these same segments can be randomly spliced. For T cells, they can undergo negative selection after this process and eliminate any self-reactive T cells.
Affinity is the strength in the binding between one antibody binding site on the antibody and one epitope on the antigen. This can be expressed by Kd, or the dissociation constant, which is the molar concentration of an antigen that is needed to occupy half the amount of total antibodies in a solution. When the dissociation constant is higher, the affinity is going to be lower, or vice versa. Most antibodies in the primary immune response have moderate affinity at around a value between 10 to the negative 6 and 10 to the negative 9. In secondary immune response, B cells begin selecting antibodies that are better binders, meaning that B cells with higher affinity will survive and replicate, and the Kd value decreases between 10 to the negative 8 and 10 to the negative 11. This is known as affinity maturation, which only occurs in BCR, not TCR. Those remain fixed for the entirety of the cell life. The development of lymphocytes begins in the bone marrow, where hematopoietic stem cells differentiate into common lymphocyte progenitors. This is where they choose the path of either becoming a T or B cell. In early lymphocyte development 3, this is where selection is going to begin. Pre-B cell receptors or pre-T cell receptors are deemed to be functional by testing it with only one chain for each, the heavy chain for B and the beta chain for T. If the receptors work, they will continue on in their development, and if not, they will die by apoptosis. Another form of regulation is going to be positive and negative selection. This occurs when the formation of BCR and TCR is now complete. Positive selection consists of a weak or moderate recognition of self-MHC molecules, which ensures survival. Negative selection is going to be the recognition of an antigen receptor being too strong, which will activate apoptosis. Negative selection is used to combat the progression of autoimmune diseases due to the faulty cell receptors. A way in which this is regulated is in the development of lymphocytes. If the binding between an antigen and antibody is too strong, the cell will be eliminated. This is going to ensure that the receptors are self-tolerant, meaning that the receptors will be able to differentiate between foreign pathogens in the body's own cells and tissues. Cells that strongly recognize self-antigens are removed before they can enter circulation, preventing potentially harmful self-reactive lymphocytes from becoming active in the immune system. If this recognition does not occur, the receptors will begin to attack healthy self cells, which can lead to autoimmune disorders. Without negative selection, the immune system would have difficulty distinguishing from self and non-self, leading to harmful immune responses.